Hi, and welcome into our talk about glycolysis or the glycolytic system. Before I get into this, I would recommend that you watch our talk about ATP, like what is it, and the first two forms that we use to support increased need, which is the stored form, like just the amount I've stored inside of a cell, and the creatine phosphate system. So for this one, we're going to talk about glycolysis, or lysis means the breakdown, and this is going to be the breakdown of a glucose or a glycogen. So the first thing that we do here is inside of our bloodstream, hence why it's red, we have these glucose molecules, which is just the simplest form of a carbohydrate that the cells really like to use. And we're going to pull it in through this black line, which is the cell membrane or the wall of the cell. So once it's inside of the cell, we're going to actually use, so I'll put minus 2 ATP to do what we could call priming it. And before I go further, be aware there's a lot of steps I am not including here and a lot of enzymes that I'm not including here because until you go into physiology or biology, you really don't need to know them for our purposes. So we're going to actually use two of these first to do what we call priming. And at this point, we've got one of two fates. Fate number one is that we take this glucose and we turn it into glycogen, which is the storage form. This stuff is good because it's a form of a carbohydrate we can store inside of a cell. When you recover from a day of, let's say, training, running, whatever, your cells are going to make or remake this stuff and try to fill up their stores. This is part of the idea of a carbohydrate loading is to increase glycogen storage. So that's fate one, and we'll come back to how it's used in a second. Fate two is it continues forward into what we call glycolysis. So we're going to go through a couple steps, and I'll just put glycolysis here. This is a process where we're going to actually fully go and break this down as much as we can. If I'm a glycogen and we want to use this, it also goes into this as well. All right, so they all go back into this exact same form here. The result of this is we're going to gain a bunch of stuff. First one is for every molecule of a glucose or a glycogen that goes through this, we're going to gain a total of plus 4 ATP made, which is really pretty minimal, and that's not really the point of why we're doing all of this work. This is just a nice, basically bonus for us that we can go and make right here. We're also going to put plus 2 of these things called pyruvate, which will go into the next step, which is the next talk here. We're also going to make, last thing here, plus 2 of what we call a proton carrier, which is NADH. This right here has the potential to become a lot of ATP. We just have to get it further down into our cell. So important facts for you, and I'm going to switch to blue here. A few things I would go and notice. First of all of this is occurring directly inside of the cell, inside of its kind of like fluid-filled center. So I can do a lot of this because, relatively speaking, there's a lot of this space in there. Part two, one important thing is if you look through this entire cycle, you'll see no oxygen needed. This is really good because if I'm at the point where I don't have enough O2 available, which we'll talk about later as we talk about warm-ups, I can run this cycle right here continuously, and I don't have to worry about having O2. There is a downside though, and that downside is this pyruvate has one of two fates. Either we're going to go to the mitochondria, which I'll just put mito, which is on our next talk, or we're going to have to go and convert it into lactate. All right, these two fates here are going to be our next two talks, so we'll go and talk about the aerobic system in one talk and lactate and why I care about it in a different talk. So go ahead and feel free to go and jump ahead onto those ones. Again, if you have a question about this stuff, bring it into our practice, and I'm more than happy to talk about this. See ya.